Mitosis video for Lab Group 3. Interphase is the beginning of the cell cycle. It is the time period where the cell spends most of its time. During this phase, the nuclear envelope increases its surface area. One thing that can be seen during interphase is two centrosomes, which make up centrioles in animal cells. In S phase, the chromosomes grow and duplicate to get ready for mitosis. The chromosomes at this point are made of uncondensed chromatin, as you cannot see the chromosomes yet. In prophase, the chromatin condenses, forming chromosomes. Also, the nucleoli disappears in prophase too. Each chromosome is made up of two identical sister chromatids, attached together by a centromere. The mitotic spindle is formed by the centrosomes. They are used in metaphase. Also, there are asters coming from the centrosomes, which are small spindles facing the opposite direction of the big spindles. The spindles are made of microtubules. Prometaphase is the phase where the cell gets ready for metaphase. Inside the cell, nuclear envelope fragments can be seen, as this is the phase where the nuclear envelope breaks down. Then, microtubules extend from each of the centrosomes at each side of the cell can go into the nuclear area and reach the chromosomes. Now, each of the two chromatids of each chromosome have a kinetochore, which is a specialized protein structure at the centromere. Microtubules that attach to kinetochores are called kinetochore microtubules, and ones that don't connect are called non-kinetochores. These non-kinetochores interact with those from the opposite poles of the spindle. After the kinetochores are attached to each sister chromatid in the mitotic spindle, the longest stage of mitosis is called metaphase. Metaphase begins. At this stage, the centrosomes are now opposite poles of the cell. Equidistant from the poles in the metaphase plate, a plane where the centromeres and chromosomes lie. Each chromosome's sister chromatids are attached to kinetochores that are attached to kinetochore microtubules of the spindle coming from the poles. Next is anaphase, the shortest stage of mitosis, which only lasts a few minutes. Sister chromatids become individual chromosomes as they are separated when the protein complexes that held them together are cleaved. The chromosomes then move to the poles as their kinetochore microtubules shorten. They move centromere first, as that is where they are attached to the microtubules. During this, non-kinetochore microtubules lengthen, elongating the cell. By the end, the two ends of the cell have equal number of chromosomes. Mitosis ends with telophase, where nuclear envelopes form from parts of the parent's nuclear envelope and other parts of the endomembrane system. This forms two daughter nuclei with nucleoli. Mitosis results with two genetically identical nuclei through the division of one. After mitosis, cytokinesis occurs. Cytokinesis is the division of cytoplasm. In animal cells, a groove in the cell surface powered by actin microfilaments and myosin called a cleavage furrow forms. This pinches the cell in. In plants, however, vesicles colocase produces a cell plate. This plate enlarges until its membrane fuses with the plasma membrane, forming two daughter cells with a cell wall in between. In both cases, mitosis paired with cytokinesis results in two identical daughter cells with the same genetic information in their nuclei.